Right, so great. So this video is based on a popular request. We want to look at the fundamentals of bioinformatics, the what we need to do if you want to engage in bioinformatics studies. Let me try and see if I can get if there is a share button so that I could share my entire screen. Okay, I'm unable to get a share button. I wanted to share my entire screen so that I'll be able to demonstrate how this works. Okay, so bioinformatics simply means that you are combining different disciplines to be able to understand the genome of any organism, or to be able to understand how genes code for proteins. And so, because of that, there are other things, there are a lot of things you need to know, which are quite fundamental. And for you to be able to know that, you need to know uh, the various aspects of bioinformatics and what it entails. Before I will talk about that, let me say that we have, the various disciplines that is made up of bioinformatics: the biology, the IT, mathematics, and statistics. These are the various, aspect of bioinformatics. Now, but there is a general principle that you have to follow. What is that general principle? In bioinformatics, there is the aspect which is wet lab and then the dry lab. The wet lab is actually made of how you are able to extract your genetic material from any organism. It can be plant, it can be animal based. I mean, after extracting the genetic material, which can be a DNA in the case of eukaryotes, it can also be an RNA in the case of like viruses and stuff. Now, after the genetic material has been extracted, and for the purpose of this video, I will restrict myself to only the DNA. Now, after the DNA has been extracted, the next thing is to make sure that you prepare something which is called a library. The library is just like a solution, and then and the purpose for preparing the library is to enable your machine to be able to read how this DNA molecule is being arranged or is being packed. So now the library preparation is being made, and then it's in the form of a solution, or let me say a liquid. This liquid is being shipped into a particular device, which is called flow cell. The flow cell is inserted into a machine which is fitted with laser light. The laser lights begin to flash on the solution, which is made of the DNA. And when it flashes on it, it reflects different colors. And those colors is what allows us to know whether it is a thymine, it is a guanine, it is cytosine, or it is adenine. These are the four types of DNA that we can talk about. And now, immediately it reflects that light, then it records it accordingly. This process is what we call sequencing. And now, the process of also reflecting this light is what we call reading. So when it has read the whole thing from beginning to the end, it is called a read. So it has read the whole thing. Now, let's say it's reading 150 bases at a time. And so when it reads up to 150 bases, that set of reading is what we call a read. Then the read is there. Then it will come and start reading again to another set like that, and that one's also called a read. At the end of it, we have reads in which represent which represent the entire genome, or which represent the genome that we are trying to determine the sequences. Where we get confused in bioinformatics is these small terminologies like sequences, sequencing, reads, and whatsoever. I've just told you that when a DNA is extracted and then a library is prepared, the library is simply like, let me put it in a very layman's language, like when the DNA is being put in a solution form, which is actually a solution though, then A machine will have to be able to read how that DNA is packed. Remember, the DNA is found within the nucleus of every cell. Not every cell, anyway. It's found in the nucleus of a cell. Now, because it is found there, it's actually a large molecule. This molecule is full of smaller monomers or smaller DNA particles called what are nucleotides. But how are we going to know whether the nucleotides are packed in this format or that format? Which nucleotide comes first? Which one followed next? Which one followed third? And then in that order. That is why the sequencing comes in. So the sequencing machine is able to tell us how they are arranged. The whole molecule, the particles have been arranged. And so the machine can read a particular length at a time. That length that the machine read at each time is what we call a read, like the ordinary read. And so the process of doing the reading is what we call sequencing. The output of the sequencing gives us sequences. So which means each nucleotide is a sequence. Another nucleotide is a sequence, another nucleotide is a sequence put in a series, just like we have in mathematics called sequence and series, put in a particular order. Now, after the sequencing has been done, now the machine has read it and it has packed it into a different file. That file is called a FASTQ file. And now, when you have your FASTQ file, the first thing you need to do in bioinformatics is to check whether the quality of the file is good or not. Because from the process of extracting the DNA to the process of doing the sequencing, there are mistakes which are always made. There could be contamination, there could also be some impurities. It's possible that some of the laser machines was not properly fixed, and then it's possible that some of the bases are not properly called. So this is the reason why we do quality checks. The quality checks then allows us to know that, well, these are the number of bases that were not properly called or were not properly read. If you ask me of what properly called means, I can explain. Now, when you do the quality checks, you will know that some of the reads, I've already explained reads, may be bad reads, or some of the bases, like the sequences, may be bad sequences. And then it allows you to be able to clean the data. And remember, in every aspect of science, there is something about quality checks and there's something about quality assurance. In fact, even in publications today, there is quality assurance. At universities today, there is a quality assurance office. It's important to always have quality and check quality. The same way with DNA analysis. So after you've done the quality checks, you use there are devices which are which we call them bioinformatic tools, in which we use in doing all these things. The DNA extraction that I mentioned, you would have used the DNA extraction kit, like the Quagin and whatsoever. They are all there. Now, when it comes to the reading or the sequencing, there are a lot of sequencing machines. We have and there are different platforms and then different sequencing machines. There's a difference between the sequencing platform and the sequencing machine. The sequencing platforms are the whole company which have set up with the softwares and the machines. So when you buy their machine, you use their software. So to get the software together with the machine that the company has manufactured, together with their website and their database as to how you are able to do your sequencing and whatsoever, is what we call the sequencing platform. So we have the Illumina sequencing platform, we have the Nanopore, that's the Oxford, and then whatsoever. Now, when it comes to the sequencing machine, we have the Mini Ion. There are quite a number of them. I can't mention all of them. Now, after you have done that, then you come to the quality. The sequencing has been done. The reads are out. It is out in a FASTQ file format. It's out in FASTQ file format. Now, you've done the quality. How are you going to do the quality? You use a tool called FastQC. If you have that tool, you have to install it. It works in both R and then Linux environment. So you check your quality using the FastQC, or you can also check your quality using the multi-QC. Now, after the quality has been checked, if there are some B's or some bases that have to be taken out, you have to also look for other tools to be able to do that. And that's what we call the data cleaning. To clean the data, to remove those bad reads and then those bad sequences. Removing bad sequences, we call it trimming. Removing bad reads is called filtering. You can use one tool to achieve the two. The most common one is Trimomatic. There are other tools like Sickle, in which you can use, but each of them works on a particular environment. It can work in R environment. It can also work in Linux environment. But Linux is always the best. Then you use those ones to be able to clean your data. Now, the FASTQ file is clean, and then it is ready for the next step. The next step is to do what we call reference mapping or genome assembly or de novo assembly. Let me give you the differences. Reference mapping, it simply means that the time you were doing the the DNA, you extracted your DNA and you were going to do the sequencing. I've already said that DNA is a large molecule. So there was no way you can sequence the whole genome from beginning to the end. So you would have fragmented the DNA into several pieces using your primers, and then you would have cleaved some adapters to each of them before you do the sequencing. Now, because you have fragmented them, after you have finished cleaning, you need to stitch them together. Now, stitching them together to form a complete long genome for a complete analysis is what is called the reference mapping or the genome assembly. De novo assembly, you don't have a reference genome, but reference mapping, there is a reference genome for which you have to make sure that you match your fragment with the reference genome to be able to do the stitching, to stitch all those pieces together to form a complete genome. And the reason we are using reference is that there might be some portions of your genome during the sequencing which some bases are missing. The reference genome will allow you to fix those missing nucleotides or missing bases. And now, after you have done that, it means that your genome is now ready. You have done your quality, you have done your trimming, you have done your assembly. Now, when you have assembled it, it turns into a different file format called FASTA file. Now, with this FASTA file, there is another quality check that can be done there, even though we always omit that portion. It is called the check M completeness and the check M contamination. You can check for this kind of quality. And what in this case you are simply checking is to find out whether, even though this is actually a quality assurance step, not quality checks step, it's quality assurance. It's done mostly in GWAS. Okay. And so now at this point, let me say that you have done your quality checks, you have assembled your genome, and then it's now a FASTA file format. At this point, the next step in your bioinformatics work is to now know, where am I going next? Where you are going next depends on your research objectives. It depends on what you are looking for. If you are looking for genes, then it means you will have to do something we call annotation. Then there are a lot of annotation tools that you can also use. But it's like the reference mapping, or the genome assembly, I didn't mention some of the tools. There are quite a number. You can use barrow wheel alignment like BW. You can also use SPADE, that's for de novo assembly. You can use Abyss, it's also an assembly tool. There are quite a number of them that you can use to stitch them together. All right. Now, after you've done that, then you get to this particular point. From the point I was talking, from the wet lab processes to the dry lab processes up to where you have assembled the genome, that one is basic for almost every bioinformatics processes. Now, from at this point, what is the next thing? It depends on the objective of your work. What are you doing the analysis for? If you are looking for some genes, then that's what I started with. It means you have to do what we call annotation. The annotation would require that you are trying to define the gene regions of the genome. So you do what is called annotation using a tool called PROCA. You can also use something called Rust. That's for reference annotation or functional annotation. And then when you have finished your annotation, then other files are going to come up like GFF files, like FSA file, FNN file, and then all of them. Or you can also have the GPK file. Supposing you want to check for, let's say, genomic islands where antibiotic resistant genes are deposited, then you may want to use the GPK files and try to do this in other tools to be able to find out that whether the genetic islands are there or not. Supposing you didn't want to go through this site, but you wanted to just find out whether the proteins are related or not, you may want to now do alignment from this point. So right after your FASTA file, if you are going to do alignment, you don't have to multiple sequence alignment, pairwise alignment, global alignment, whatsoever. You don't have to go through that side of like trying to do annotation. You simply just go and do your alignment, depending on the type of alignment you want. If they are just two genomes and you want to just compare them, maybe you may want to do a pairwise alignment. If it is a short gene and you want to you want to map it to other reference genome to find out whether the protein is contained there or not, then you are doing a local alignment. I've explained these things in a different video. And then what is it? You are good to go. Supposing you wanted to do a phylogenetic tree, you wanted to do clustering, then you have to do an alignment first. The alignment will allow you to be able to generate other files like the NUWIC file, which you can use to visualize your genome in the form of phylogenetic tree or in the form of clustering. You may also want to extract SNPs. And then at this point, when you have gotten your FASTA file, you can also go ahead and start extracting your SNP from there. There are a lot of tools which can be used to extract your SNPs. Maybe you want to do SNP analysis. So you can extract your SNPs from that place. Then you go ahead and then start your analysis from there. So I will summarize it here. From the beginning, from the wet lab to the dry lab, up to the assembly or reference mapping is practically a general thing you have to do in bioinformatics. Right after that, you will now branch to an area based on whatever that your attention or your objectives are. If you are going to go into RNA-seq or like gene expression, if you are going to go into GWAS, if you are going to go to like genome-wide association studies, if you are going to go into CMV, like copy number variation, those who want to check for variant or want to do variant calling, then your objective will not tell you the next thing you have to do with your FASTA file, and then you can take it up from there. It's going to allow you to extract other files. For instance, if you want to do something like, let's say, RNA-seq, the files in which you use for RNA-seq and the tools vary. So you will now, from there, know the kind of tools that you require to be able to check, to get the genes, and then to be able to examine the expression level of each gene. That's what RNA-seq is about. It's about gene expression. And that is how bioinformatics work. I think that I've tried to answer the questions that I was supposed to do, but it's rather turned out to be a long video. But this is the general principle. Now, of all that I have spoken about, bioinformatics works on its own, like I defined earlier. You take this is the biology part of it, but to be able to do what I have done, you require some kind of knowledge in IT. In IT, what is the area that you need? You need Linux. You need to have complete sense of Linux. It means that at each step, if you are going to have come out from the wet lab, you have done all the sequencing, you should know that you are supposed to do FASTQ. You need to do this in Linux environment. What tool is required? You require a FASTQ-C. Then you have to make sure that you master your Linux environment. You know how to create folders. You know how to create files. You know how to move files around. Or you know how to run the code in Linux environment and how to get your output. There are instances you may have to work on a server because if the data is large, you can't deal with your local machine. So you should know how to move your data into the server, work from the server, and bring the results out back to your local machine and begin to work from there. That's where Linux come in. Some of the output, you may want to visualize it, and then you may require miscellaneous tools like tools like R or like Python to be able to do that. And so when you go to the Linux environment too, before you can install all the tools I have mentioned, like the fast QC you want to do, there might be some dependencies. You need to also install those ones. Sometimes you require something like Java to be installed. You require something like Python, like the if you want to install the tool, and the tool is a Python tool. So like pip install. Then if it is a sudo, you have to make sure that those ones are installed. If it is Anaconda tool, you have to make sure you have Anaconda also installed. And of course, most of them work on Java environment. You have Java also installed. Even if you are going to install the tool, which is a window interface user tool, you still have to have some of these things installed. If not, they will not work. Something like bricks, which is used to do, which you can use to compare ORFs, like open reading frames. You can also do to compare genetic islands. If you don't have Java or installed on your computer, you can't install it. And so this tools installation is based on your objectives, like I've already explained. You should know at which point you are supposed to install which particular tool and what that particular tool is supposed to be used for. But in all cases, you need to master Linux environment, know how to go about Linux environment. Very important because much of the work will be done in the Linux environment before the output is then brought to R or to Python for visualization, or even to Excel, Google Sheet, or Google Colab. All of them, SPSS, all of them can now be used to visualize the output. But then the Linux environment tool can also visualize some of them, but you still have to bring it out to be able to have a clearer view. Okay, so I will leave it here, and I hope that I've answered the questions that I promised to do. Thank you. See you some other time.